Okay, so how do all of those things that we just talked about, the MHC complexes and cell mediated and humoral immunity come into play, or how does it kind of act out? Okay, so the first thing you sort of need to know is that T cells, which are really the soldiers of the immune system, are not able to recognize some sort of bacteria just kind of floating around in your body or virus or anything like that. They can only recognize what they've been presented with. And so there's an entire class of cells called antigen presenting cells, or APCs for short. So these antigen presenting cells, they're going to be dendritic cells, which have nothing to do with neuro stuff. They just have like a lot of branches. These are going to be macrophages and B cells. So what do these APCs do? Well, let's draw one of these. Let's say we've got one of these dendritic cells running around in your body, and it has the MHC complexes on it. And it also has receptor sites. Let's make those red for different things. And let's say a bacteria comes along and it will attach to one of the receptor sites. And this dendritic cell is going to pull that bacteria into its cytoplasm. And then it's going to chop it up into little pieces. And then it's going to take one of those little pieces and present it up here on its MHC complex, sort of like raising its hand saying, hey, here's a thing, and it could be a bad thing. So if this thing is from the outside, if it's an exogenous antigen, then this MHC complex here is going to be an MHC2 complex. If it is an endogenous, if it's a cancer, if it's a virus, viruses, they kind of make our cells go haywire. So our cells know to chop up the parts that are making it go haywire and present it on the outside. And in that case, it uses an MHC1 complex. So we've got this, so we have this presented antigen right here, this epitope here. So what's next? So we've got something, it's been digested, we've got a piece of it hanging out here on this antigen presenting cell. So what now? So this is where T cells come into play. And I guess I'll make T cells purple. So when a T cell comes along, with a receptor with its own site that matches this antigen. And it also, so there's the matching receptor site. It's also going to have a little helper called a CD4 complex. And so when this receptor site and this CD4 complex work together, this T cell here becomes activated. What does that mean? Well, one of the things it means is it sends out these sort of chemical signals to other and kind of recruits other T cells, and also as a positive feedback loop, it'll kind of magnify the signals. This T cell will also go what's called clonal selection. And so because of these chemical signals, it's going to start making copies of itself with that specific receptor site and the little CD4 helper. I dropped my lid. It's going to start making clones so it can get more help in activating. Okay, so if a T cell is activated by, so we call this, so this is an MHC2. If this were an MHC1, the T cell that might attach would be a CD8. So if it's an MHC1 complex, that means something is going wrong from inside the cell. So these CD8 complement ones are called cytotoxic T cells, and they are really the ones that can actively destroy bacteria, viruses. They let out the sort of toxic stuff that really disables the pathogen. So that's sort of a very brief overview of the CD4 and CD8 T cells, as well as the antigen presenting cells. So let's just skip over here and do the antibody mediated immunity really quickly. So let's go over here. We'll do antibody mediated. And it'll be blue for B. B for B cell. So we also have receptor sites on these B cells. And when we have something that fits on that receptor site, a bacteria or something, it will also undergo making clones of itself. And when a B cell is activated, it will make plasma cells. And what these plasma cells do, so here's plasma, is they're basically antibody factories. And so they're going to be spitting out all these antibodies into the fluids, into the fluids of the body. And these antibodies themselves can start to cause trouble for the pathogen, along with setting off this CD4 kind of complementing what these T cells are doing over here. These antibodies that these plasma cells are pumping out are going to recruit what's going on over here as well. The other thing that that the B cell clones into are memory cells, and they are going to hang out and live for actually years. Some of them are thought to live for decades. And so these are the memory B cells. So these antibodies, we talked about how the antibodies themselves can cause trouble for pathogens. So they really are kind of shaped like a Y. And they're proteins. They're made out of proteins. This is sort of a cartoon version. Proteins, as you may know, are three-dimensional in structure. They sort of wrap around themselves because of the hydrogen bonding and the interacting that goes on, sulfide bridges and salt bridges that happen that give them a very individual shape. And depending on the order of those proteins, that's how that shape can change. So this part here, this is called the heavy chain. And then you have this other part here called the light chain. And this part here at the top, and over here too, this can be variable. And just how I was talking about how if you swap a different amino acid, it's going to change the shape of that protein. Well, in this variable space, again, you have that idea of variation leading to specificity as well as ubiquity. So you're going to have lots and lots of different variations, but also they match very specifically. So these antibodies, they have sort of different classes. And another name for antibodies is immunoglobulins, because they sort of, while they do have a somewhat Y shape, they're kind of globular as well. If you can think of them existing in liquid and sort of the hydrophilic parts being on the outside and the hydrophobic parts being on the inside. Anyhow, they're shortened by IG, a capital I, lowercase g. 1, 2, 3, 4, 5. And then they have a big letter on the end that sort of classifies them further. So IgGs is the most abundant. They're found in your blood. They're found in your fluids. And this one is that one that we talked about earlier. It can cross into the placenta and confer immunity to the fetus. We have IgA. There's quite a bit less of this one, and it's going to be in like sweat, tears, and also quite importantly, breast milk. Again, covering those babies until they're able to do this stuff for themselves. IgM, these are kind of the first responders, honestly. When those plasma cells that we talked about or those B cells become activated and become plasma cells. These IgMs are the first line of antibodies that are put out. And actually, I'm going to draw a little picture of what these look like over here on the side. And these over here, the IgAs, are kind of double-sided. And you can see why that would be a good thing, because you can have not just 2 sites here, but you can have 4 sites where you can start gumming up the works. These IgMs, so they're like first responders. And so if someone has these IgMs, a bunch of IgMs in their blood work, then it's very likely that they've had a recent infection. And these, you may have to bear with me. There's actually, there's 5 all stuck together. 3, 4, 5. They look kind of cool, like a snowflake, right? IgD, these are really small, I think less than 1%. Most of the places you find these are actually as receptor sites, initial receptor sites on B cells. And then finally we have IgEs, which are even smaller proportion as far as percentage of antibodies in the blood. However, these are also just a normal sort of Y configuration, but they are associated with allergic and hypersensitivity reactions. So they are associated with mast cells and the histamine response. And so that's IgE. So even though there are not very many, they are really quite powerful in eliciting response from the body overall. And I think that takes us all the way through immunity. Yeah, thanks for sticking with me to the end and late at night, or it feels late for me because all my kids are asleep. All right, have a good night and study hard, and I'll see you tomorrow.